Hello, everyone, and welcome. My name is Shannon Tyler, and I'm the Teacher Enrichment Program Manager at the Center for Excellence in Education. I'll be your host for today's webinar titled Step into STEM, Junior Innovators Challenge. In today's session, we'll be hearing from a student researcher whose work earned national recognition through one of the country's premier STEM competitions for middle school students. Our speaker, Pranshi Mehta, will share her journey from developing an idea to competing in the Thermo Fisher Scientific Junior Innovators Challenge, including insights into her research, the competition experience, and how students can begin exploring STEM research opportunities of their own. Whether you're a student interested in science and innovation or an educator looking to encourage research and curiosity in the classroom, we hope today's session leaves you feeling inspired and empowered to take the next step in STEM. With that, let's go ahead and get started. Before we begin, I'd like to briefly introduce the Center for Excellence in Education, or CEE. CEE is a nonprofit organization founded in 1983 with the mission of helping students develop the knowledge, skills, and confidence to pursue excellence in science, technology, engineering, and mathematics. We provide cost-free programs for students and educators that emphasize advanced STEM learning, research experiences, mentorship, and engagement with experts across STEM fields. I'll briefly highlight our programs on the next slides. Two of CEE's flagship programs are the Research Science Institute, or RSI, and the USA Biology Olympiad, or USABO. RSI is a highly selective, cost-free summer research program hosted at the Massachusetts Institute of Technology, or MIT. The program brings together talented high school students from around the world to conduct original research alongside leading scientists and researchers. Students in RSI gain hands-on experience with the full research process, including reading scientific literature, designing experiments, analyzing data, and presenting findings. The USA Biology Olympiad is the nation's premier biology competition for high school students. Through a series of rigorous exams, students deepen their understanding of biology and may advance to the national training camps and international competition. Both programs encourage students to challenge themselves academically, pursue scientific discovery, and connect with a global STEM community. CEE also offers the Teacher Enrichment Program, or TEP, and STEM Lyceums, which are the programs bringing this webinar to you all today. The Teacher Enrichment Program provides professional learning opportunities for educators, helping them bring advanced STEM content, research opportunities, and innovative teaching strategies into their classrooms. Through webinars and additional resources, TEP supports educators in fostering curiosity, critical thinking, and real-world problem-solving among their students. STEM Lyceums is a virtual program designed for students to explore STEM careers and connect with professionals and researchers from a wide range of fields. These sessions give students the opportunity to learn directly from experts, ask questions, and discover new pathways in STEM. Together, these programs help connect classroom learning with authentic STEM experiences for both students and educators. Before I introduce our speaker, I'd like to go over a few quick Zoom norms. I encourage you to participate throughout the session by asking questions in the Q&A or chat as they come to mind. You do not need to wait until the end of the presentation to submit them. I will reserve a time for a Q&A session following the presentation and will answer as many questions as possible. A recording of today's webinar will be shared with all registrants after the session. And with that, I'm happy to introduce today's speaker. It's my pleasure to introduce Pranshi Mehta. Pranshi is a student researcher exploring how biology and machine learning can come together to tackle real-world health challenges. Her work focuses on using artificial intelligence to support antibody design for complex diseases, and she has shared her research through global science forums and public conversations. Beyond research, Pranshi is deeply engaged in mentorship and STEM advocacy, where she emphasizes that meaningful science starts with curiosity, persistence, and the courage to ask questions. As the 2025 Thermo Fisher Scientific Junior Innovators Challenge First Prize Science winner, Pranshi is excited to connect with students and show how small first steps can lead to big discoveries. We're excited to learn from her today, so please join me in welcoming Pranshi.
Thank you so much. Hello everyone, my name is Pranshi Mehta and I'm super excited to talk to you guys today about stepping into STEM and the Junior Innovators Challenge and more about my journey. This is the agenda. I'll just start with a brief introduction and then going to the bulk of the presentation on research. We'll start with the why and then getting started on research, actually conducting the research, taking it to competition, and then I'll finish off with some more details about JIC in particular as the nation's premier middle school science competition, and then I'll finish off with some conclusions and advice. Here's just a little bit about me. Outside of school and research, I play piano, I sing, I run, and I bike. And then on the more academic side, I do math counts, science bowl, robotics, and then I really love to write, so I published a book when I was 10. So diving right into the research, I think every great research starts with a why. That is the starting question for the research, and it's the main driver. It's where your idea came from, and I think it can be more like an everyday moment or a problem that caught your attention. You have to talk about why it matters to you personally and how you have a connection with this, and this is what drives you throughout the entire research. I like to call it like the spark and passion, because I think it's what really motivates you to continue throughout the research. I think that this is really important, especially when it comes to competition, because it shows how much you care about the topic and why you did the research that you're conducting. I think along the lines of the why, you have to narrow it down from a very, very broad idea to something more testable and manageable, and I'll get to more of that on the next slide. So getting started. I think the biggest part of getting started is taking your vague curiosity and turning it into more of a researchable question. So making this big idea that you have more testable. So if you have something like air pollution, maybe you could narrow it down to certain things that you see in the air or air quality, or maybe a specific location versus just air pollution. Then you could do background research. So what's known and what isn't known, and basically just trying to become an expert on the topic that you have at hand, I think is really important, so that you know what there's still left to find out, and you can do something novel and new and creative in the field. Then you start to design the experiment. You find the materials, the setup, and the variables. I think that one thing that's really misunderstood about designing an experiment is that it's set in stone. A lot of times people say that you have to have this strict plan and you have to follow it the entire time while you're doing your research, and I think that especially with my research, that was not the case at all. While I did follow the same general roadmap of my original design, I edited and tweaked a few things along the way, so my design was more of a working document versus something I made at the beginning and then followed throughout the entire time. And I think that it's really important that this remains flexible, because things aren't going to work the way you want them to, and it's really important that you're able to shift your experiment to go the way that works. I'll talk a little bit more on my research now and how I align with these two first steps. So starting off my why was my friend's mom. My friend's mom passed away due to cancer and brain cancer. So when I realized this, I would watch her come back from all these treatments that wouldn't really do anything. And I wanted to find a new cure, or at least a novel up-and-coming treatment method that could be applied to brain cancer, because there were all these old treatments that clearly weren't working, since there was still more deaths occurring. So when she came home one day, I started researching on the Stanford AI and Stanford Medical Journal website. And I think what's really fascinating about the intersection of these two is how many new cancer treatments it creates, because I came across something called CAR T-cell therapy. And CAR T-cell therapy works really, really well for liquid cancers like blood cancer, leukemia, etc. But it hasn't been applied to solid cancers yet, and that was what my entire project was about. And so that was my why and my spark. And then when I started getting started, a lot of the tools I used were all online. And the material and original setup I had required me to use Haddock, but when I tried to use the tool, it didn't work. So I switched over to HDoc, and I think that really goes to show how in every experiment, you cannot just have a set in stone path, and you have to move it to the way that the experiment's going. Now, the last bullet point, find resources or tools. Now, not every tool has to be in a lab, and I think that this is a really big misconception, that in order to do research, you have to have like a fancy lab and all these fancy equipment. My entire project was done on my computer by myself, and I did all online databases, government databases, AI tools, like I mentioned from Stanford AI. So I think that it's really crucial that the kids today understand that you can do whatever you want from your computer, and it's getting even bigger with the age of AI, that you can search something up and get a tool, and you can use it on your computer from your desk. You don't need to go to a lab for all of this. So I think that's another big thing, is to figure out what resources or tools you're going to use, even if it's online, so you at least have the plan in front of you. And again, this can change. The next step is conducting the research, and this is on the day-to-day basis. It can be even 10 to 30 minutes a day that you just come home and you do your research and conduct your research. And I think that another benefit of having all these online tools available is that the research you do can be put on pause. For example, in a lab, you would have to go there for hours and dedicate so much time to finish up your experiment fully because you can't leave things overnight. But online, you can just hit save or you can hit pause and everything will stop when you're ready to come back to it next time. And that's also what makes these online tools, when you're conducting your research, more efficient and easier to use. Some people call this the most boring step, but in my opinion, this is the most important step because this is where most of the work actually happens. You get all your charts and your data and your graphs from this step, and it's really important for you to analyze this and to get the results as well. I think an important part of this step is to keep going because it's going to get hard and you're going to hit a roadblock eventually. Like I mentioned, I'll go more into depth. I was going to use the tool haddock in my research to dock these two proteins to each other. And that was one of the main tools that was in my materials list. But then when I started using the tool, it was really slow and it started lagging a lot. So that's when I had to make a hard left and turn over to HDoc, which was from like a Chinese lab. And I found that that tool was much quicker and much easier to use, but I wouldn't have found it if I hadn't kept going and gone back to my spark or passion of why I started the project in the first place, which is my friend's mom. And I think that your spark really comes useful here because it's what reminds you to continue doing your project and doing your research, even when the times get tough. And I think another thing I like to remind here is that wrong results are still results. Even if something doesn't work or it's invalid or you get a theory that it doesn't work, it's still a result. It doesn't mean that you should just throw it away and like start over because it still tells you something. And I think this is really like the Thomas Edison quote. I think I didn't fail, I found 100 ways that don't work really goes to show that just because it doesn't work doesn't mean that it's a failure. It's telling you new information every single time. For example, I'll go more into the process of my research here. The way I did my CAR T cell therapy was I found an antibody that could dock to the cancer, and it would dock in the correct spot and it would stay attached very strongly. And I would test these antibodies over and over and over again. And after 10 of them, like none of them were working. And I started to get really discouraged, and I would remember that just because they don't work doesn't mean I'm not making progress. I'm still making progress by figuring out what's not working. And then I started testing more and more, and I eventually found the one that did work. I think the next phase after you conduct your research would be taking it to competition or to the science fairs. And the two main parts of presenting to a science fair that I like to break it down to are passion and preparedness. First, passion. You should show whoever you're presenting to, the judges, your parents, your friends, how much you care about the topic. And I think this would go back to your why or your spark. You need to show them that you truly care about what you're researching and that you want to learn more, and that's why you started the project in the first place. You should show that that was the driving factor. And then second, you should show how prepared you are. So tell them all about your background research. Tell them all about what you did throughout the project, because I like to remember that judges only judge what they see on your board. So you have to make sure your board is visually appealing. It has all the charts on there, all the data, because at the end of the day, it doesn't matter how much effort you did if the judge or the person you're presenting to can't see it from when you present to them. So it's good to make sure you're articulate about it and you have the charts and the graphs and everything ready to go so that the judge knows what your project is fully about. Definitely make sure to practice for your friends and for your teachers. I think it's really important to get some reps in before you go to present to whoever you're presenting to, just so you know what you're going into. And then I'll kind of switch gears over here to JIC and how to get nominated for the Junior Innovators Challenge. I know that some people don't go to a local fair, and some smaller states you go straight to a state fair. but whether you're at your state or your local fair, I know that the numbers differ, but I'm pretty sure first, second, and third get nominated for the Junior Innovators Challenge. And then I'll just switch slides here. And from all the nominators they get, they pick 300. And those are called the top 300. And then from the top 300, they pick the top 30. And the top 30 go to DC. And it's a really, really unique experience in DC, because not only do they have a day where you get to present in front of these very, very acclaimed judges and you get to present your research to your friends, you also have a couple more days of the week where you're doing STEM challenges. And there's some like coding. I remember there was one when we did it where it was biology and we worked with like pipettes and contaminated water. And there was some on engineering and building chain reactions. And they're really cool. You work in a team of 5 people and you work together to solve the challenge that's in front of you. And it really testifies your teamwork. It shows how well you're able to communicate with others. And then you also learn so much about the STEM topic at hand. And you learn a lot about kind of the people around you and you connect with them really well. And I think that it's really unique because there's no other science fair, especially for middle schoolers, where not only do you present to judges, but you also learn STEM topics with friends, and it's really cool. And Thermo Fisher Scientific provides a lot of great materials like in the labs for you to work with. I'll also talk a little more right now about my project for JIC, the one that I submitted and presented at the Junior Innovators Challenge. I know I touched on it throughout this presentation, but I did CAR T-cell therapy applied to glioblastoma, which is one of the most malignant and aggressive brain cancers out there. And glioblastoma has a very, very high mortality rate, which is why I picked it as the cancer to apply CAR T-cell therapy to. CAR T-cell therapy works really well for liquid cancers like blood cancer and leukemia. So when I read the articles about it, I started wondering, well, why can't we apply this treatment to solid cancers? So I researched more into how CAR T-cell therapy works, and they take the T cells from your body and they attach a CAR onto it. That's where the CAR in the CAR T-cell comes from. It's chimeric antigen receptor, and the top of the CAR is an antibody, and the antibody varies per cancer. So the T cell stays the same, it's your body's T cell, but the antibody varies per cancer that you're trying to target. So I looked for antibodies that would bind to the glioblastoma cancer, and that was what my science project was about. Using online simulators and code, I tested all the antibodies that could bind to the cancer and successfully kill it. That meant that it had to bind strongly and it had to bind in the right spot. So I tested 30 antibodies, all to the glioblastoma cancer. And once I had tested them, I used the online simulator to take a picture of them so I could see that where exactly they bound to on the cancer. And then I also used tools like Prodigy to get a measure of how strongly this antibody was bound to the cancer, because you don't want it to just touch it and float off. So those were the charts and graphs that I analyzed for my project in particular. And from those, I found 3D85 as the best antibody for this cancer. So that meant that if 3D85 was on a CAR T-cell therapy, it would most likely work for glioblastoma. And I feel like the next steps for my research would be to take it to a lab, because as I mentioned, everything I did was on a computer. And it's very important that this is verified in a lab before it's given to patients. So I feel like wet lab studies would be the next steps for my project in particular. So in conclusion, just some advice I have in general would be that anyone can start. Like I mentioned earlier, a lot of the tools that are coming online are really open to whoever just has the curiosity to search something up. So no matter your age, I don't think it's a limitation to what you can do. And I'll give another example here from my specific project. I used AlphaFold 3 which is one of Google DeepMind's AI Nobel Prize winning tools. And it used to take a PhD, their entire PhD career, to figure out the 3D structure of a protein from just its amino acid sequence. And now AlphaFold can do that in a matter of seconds. And I think this goes to show how fast AI is advancing these medical tools, and so much of it is publicly online for anyone to work with. And that lowers the barrier for who could enter the medicine field, because now anyone can start research if they just have the right curiosity. And that goes to my next point, which is that youth curiosity is really important. I think youth, especially of the teenager age, have really unique opinions about the world around them depending on their environment. And it's really important. Those opinions matter a lot, if not as much as more than those of the people who are currently in the field. So I think that the youth who feel like their voices don't matter, then don't start research. And it's really important for everyone to know that their voice does matter, and that whatever findings or scientific findings that they have, they can bring it to the scientific world, and it's really important. I think the third piece of advice I have is to seek help when you need it. Help is always there for you, especially on your project. And like I said multiple times, all the online tools, but even your science teachers, your parents, your friends, everyone's there to help you and support you. All you need to do is just ask them for help, and they'd be willing to help you find something. And maybe as small as, hey, can you help me debug this code? Or, hey, can you help me look at this to make sure my poster's looking right? And they'll be everyone's there for you, so you don't really have to do it alone. And especially research, it's never done alone. There's always a lot of people who support you along your journey. My fourth bullet point would be to keep going. I think it's really important that no matter what happens, you keep going with your research, because you're going to face a lot of obstacles along the way. And it's really important that you just continue throughout your journey and keep going, persevering. Go back to your spark, which is the main reason you started your project, and that passion. And I think that's what kept me going when times were tough, was my friend's mom, and I was doing this project for her, and that's really what just kept me going. And lastly, have fun. Research is a really fun project, and it's really nice to see the result at the end when you've done all this research and you finally get your results. And it's so satisfying to see what your work has amounted to. Maybe you found something, maybe you didn't, but you worked so hard to finally get this result and all the charts and the graphs. And when you finally figure out something, it's just so rewarding at the end. And I think the entire journey is just so much fun to just come home from school and look at your results. So I remember I would come home and I would always look online and see if my HDoc had loaded yet. And I think it's just really fun, and enjoy the process of research. It's definitely something to enjoy. Thank you so much, and I'm happy to answer any questions.
Thank you so much, Pranshi, for sharing your experiences and insights with us today. We will now go ahead and move into the Q&A portion of the webinar. If you haven't already, please feel free to submit your questions using the Q&A function or in the chat, and we'll start going through them. We'll do our best to answer as many as possible. And so let's go ahead and begin discussing. So we have one question come in from Hector. It says, for research topics that cannot be physically implemented currently and is more of a concept like your research, how much evidence through simulations do you think would make a concept credible enough to show its possible implementation?
If I understood that correctly, you're asking for more theoretical research questions, how many simulations you would need for. So I don't think there's an exact number, but I would definitely say multiple, running it over and over again and making sure that you check the articles behind the sources you use. So I know for a lot of the sites that I used, I made sure to look for articles stating the credibility of these sites and making sure that they were credible. I know that I used one from a Chinese lab, and I definitely had to find a lot of articles on that one because it wasn't a government issued site, but definitely government issued databases should be very credible.
Thank you. And then just to be sure, or just to let you know, Pranshi, these answers or the questions that I'm asking right now are coming into the Q&A function in Zoom. You should be able to see them too, just so you don't have to just, remember them and listen to me read them.
Okay.
Okay, next we're going to go to a question from Mohita. What was your experience with finding mentors? What are some ways you recommend doing that? I have started a project and I'm looking for more advice.
I think finding mentors is mostly just asking around. Your science teachers, definitely, and then you can always email people if you know anyone. Connections are definitely really important for finding mentors. And online too, I think just searching things up. Google is really helpful. There's a lot of articles out there on things. But yeah, just asking around, and your science teachers or like parents, friends would be probably really willing to help.
Thanks. Next, we'll go to how would you proceed if you had a question you wanted to research but didn't feel that it was really all that groundbreaking or important to the world?
I think that any question that you want to research would be groundbreaking, because as long as you care about it, it's important, and you can really research whatever you want. It doesn't matter how important you feel it is to the world. As long as you really care about it, that's all that matters.
Agree. Yeah. And on that note, part of our Lyceum's program is a mentorship program that we are currently looking to build and grow. So in this program, we're pairing high school STEM students with professionals from industry. And so if you're interested, please reach out and we're happy to give you more information on it. Next, let's go to who helped you throughout the project. Was there any mentor to help you with the tools you used for your project?
A lot of the tools I found through other tools. Like I mentioned, my science teacher was the one who recommended AlphaFold to me. And then from AlphaFold, I found Haddock. And since Haddock didn't work, I switched over to HDoc. So I think a lot of Google searching and a lot of tools connecting me to other tools. But more specifically on how I found the tools, I would definitely recommend starting with the government databases. And I know that the NSD and the government databases have a list of sites that they would recommend. So definitely just going down that rabbit hole. And then for mentors, definitely my science teacher. She was a lot of help during this project for encouragement. Even when she couldn't provide exact technical help, she was definitely always there for me.
Perfect. We have another question on mentors. Mentors are popular today. I tried to contact university professors via emails online. Is that really the best way to get an engaging mentor? Or I guess, what was your experience with this?
Honestly, connections. I'll say, like I said in the previous question, my science teacher was there for me. But then if they don't respond to their email, I don't know if there's much you can do about that, but just connections, trying to see if there's anyone you know or your parents know that have your area of expertise that you could ask a couple questions to, or just things like that.
Perfect. Let's go to, what would you say is the biggest factor to win science fair?
Ooh. I would say probably how much you care about the topic and how much effort you put into your project, because it doesn't matter how big or fancy your project looks. It all comes down to whether you truly care about the topic at hand and whether you put effort forward to carry out the research, or whether you just did it half-heartedly. I think that really makes a difference, and it comes through in your research and your poster. No matter how fancy things look, it all comes down to what you care about and what you put work into.
And then to kind of piggyback off that question, what skills do you think are the most important for students interested in these type of competitions to have?
On the science fair side, I think the skills definitely vary per project, but definitely for project presentation, I would definitely say public speaking is really important, or just at least practicing and getting reps in with your board, because you don't want to just go there and then not know what to say to the judge. So some public speaking or just practicing.
Yeah. Perfect. And then I'm going to squeeze one of my own questions in before I go to the Q&A. You talked about having challenges during, your research journey and experiences and pushing through and persevering. What are some of the biggest challenges that you faced, and how did you work through them?
A couple of the biggest challenges I faced would be one of my tools not working. Like I mentioned earlier, Haddock was really slow and laggy, and so the way I worked through it was just going deeper into the internet for other tools I could use. And I think that that's just a good way to handle challenges in general, is if you hit a roadblock, would be to look for other alternatives to keep on going, because you can't just stop there. You should continue pursuing and persevering and thinking back to why you started the project in the first place.
Yeah, again, I completely agree. I remember in grad school myself, I had a professor tell me that, if you're not ready to quit, if there's never a point when you're ready to just quit and give up, something's not right. You have to get to that point if you're doing something that really is hard and hasn't been done before. So
yeah,
challenge is part of the process. It's to be expected.
Yeah.
Okay, I'm going to go through this question we have in the Q&A. I'm going to pick bits and pieces and just kind of ask you to speak on it. So the question is, basically, I have some spark and passion for specific categories, but they have a few problems encountering how to get started, basically. So the first thing is, what do you do if you're struggling with trying to narrow down the topic to something that's testable and realistic, not something of a large scale? Do you have any advice or, insights on that?
Yeah, I would definitely say to read more articles on it, because the articles definitely go down into niche areas, and if you see an interest in niche areas, then you can just pursue that. I remember I was going more broad cancer treatment, but then once I found an article on CAR T-cell therapy, which is a type of cancer treatment, I just went kind of down that line and I started applying that therapy. So I definitely think that searching something up and then looking through all the articles to see which ones interest you and which specific subtopics interest you.
And then after you complete that step and you narrow down your topic, where did you find the resources and tools that you used? He said, you bought up a few, like the Haddock and stuff like that. Where did you find them? And then how did you learn how to use them, basically?
Yeah, I think a lot of these tools are pretty straightforward to use, but I just Googled it. I Googled online docking simulator. I found Haddock, which didn't work, so I moved to HDoc. AlphaFold was pretty straightforward because it was a Nobel Prize winning. And then how to use them, I think they're all pretty easy to use and pretty straightforward to use. But if not, there's definitely a lot of YouTube tutorials out there on how to use these tools. So if you go scour the internet, there's probably ways to find out how to use these tools.
Perfect. I have another question. Let's see. So you also said that you use artificial intelligence and stuff like that in your work, as well as machine learning. What advice would you give to students who are interested in combining AI or machine learning and simulations, basically, with their biology or other scientific experiments?
Yeah, it's a great question. I think there's definitely a huge emerging field out there of computational biology with AI and healthcare, or AI and biology. And the way I would suggest merging it is kind of like I did here, using AI to assist in the healthcare field and find possible treatments and things like that. And I think that would be one important way. There's also AI can help find possible drugs for things, which is kind of similar to the AI finding treatments. And you can use AI to analyze and scan data. So a lot of the ways that AI is helpful in the doctor's office is also a lot of the ways AI can be used in biology research.
Okay. And then this is just kind of a personal opinion question to piggyback off of that one. Do you think AI is changing how students approach scientific discovery?
I definitely think it is. It's helping a lot to how students can approach it, because I think more students can approach it now, because with all these AI tools, there's so many more options for students to get creative and engage with STEM and engage with scientific discoveries. So now more students can make these scientific discoveries and at a much faster rate because of how efficient AI is making things.
Yeah, agreed. And I think it's going to be really interesting to see, what the future holds with that, what research looks like, and, the changes that are inevitable to come because of, what AI is bringing to the space.
Yeah, I'm really excited for it.
Yeah. I have a few more on just the JIC in general.
Yeah.
And I'll go back so we have a few more questions come into our Q&A. But talking about JIC, what did you learn from competing and presenting your work at like a national level?
I think for competing and presenting my work on a national level is that the background research that you do is really, really important. And I don't think this gets stated enough. It's more on like the actual research and the research that you conduct, but you obviously know that part. You can explain it really well because you're the one who conducted the research. But you have to make sure that you know all the background information around your topic to support your research. And I think taking my work to the national level really taught me that. And it taught me that you really need to understand everything around your project, not just the project itself. Because I was talking about CAR T-cell therapy for glioblastoma, but that also meant I had to understand the chemotherapy and how chemotherapy worked for glioblastoma and surgical treatments for glioblastoma. And I think you really need to know a lot more than just what you're doing.
Perfect. Thank you. Okay, so I'll put my questions on hold for now and go back to the chat in the Q&A. We'll start with the question in the Q&A. What was the result of your research and what was your hypothesis?
My hypothesis was, could we find an antibody suitable for CAR T-cell therapy applied to glioblastoma cancer? And the result was yes, and I did find one. Antibody 3D85 was compatible with the glioblastoma cancer, and it worked really well and was a possible solution if taken to wet lab.
Okay, how did you find time to do this research every day? In high school, it's much harder to find time, especially with sports clubs and the rigors of the hard classes. You mentioned in your presentation just spending 30 minutes to an hour every day, but the fatigue builds up and sometimes just, makes you forget about it or just lose motivation, keep working. Would you recommend doing it over the summer instead? Or maybe chunks or shifts during spring break, winter break, and then not working on it during regular normal school time? Or how did you deal with basically the stress of everything?
Yeah, I think that's definitely a really valid point, and honestly, the breaking it up over summer or breaks is a great idea. I think whatever works for you. If you know that there's like a certain part, like semester, where you have less clubs or activities, try and getting more work done then, or over summer or breaks is a great idea. The way I personally did it was that I would do a little bit of my work and then I'd let the simulation run, and then while it was running, I'd do like my other homework, and then I'd come back to it when the simulation or HDoc was done running. Or if it took days to run, then I would put it on for days and then I'd come back to it days later. So my work schedule kind of rotated around when my simulations and when my work was done for my research.
We touched on this a little bit when you talked about your challenges, but say if you're stuck with your research, how would you approach it and move forward?
I think that really depends on what the research is, but in a general sense, trying to find other alternatives to move forward versus what you're doing. I think trying to see a forest out of the trees, because sometimes when you get stuck, you think there's only one way to do it, and when that way isn't working, you feel like you just have to give up. But if you take a step back and look at how there's other ways to do this, or go back to your materials and your design process and kind of tweak it a little bit and then follow that new road, then you can really get around a lot of the obstacles in your research.
Thank you. What platforms do you recommend for learning the application of machine learning in biology? I tried Google's machine learning crash course, but that is really time taking and I was barely able to apply my knowledge.
Okay, so specifically for machine learning and biology, I'm actually not aware of many courses, like structured courses out there. Honestly, the best way I would say to learn would be through doing your own research for machine learning and biology. I learned a lot that way about tools, about cancer, and about glioblastoma and the brain itself, and then also how AI works in that intersection. I learned so much through the research. So honestly, I would say the best way would be to start the research, and I didn't really need any background information specifically from a course to do this. I kind of learned as I went through Google and YouTube as I was doing this research.
Thank you. How long did the whole research process take until you were ready to share it in science fairs, et cetera? Was it just a couple months or a few weeks, or kind of what did that timeline look like?
Yeah, for me, I kind of followed our Greater Austin Science and Engineering Fair timeline. So they recommend that you start around September, October, because the fair is in early February. So I really just followed that timeline. I got a lot of the simulations done in November-ish, and then I did my heavy data analyzing in December over winter break and things like that. So that was my main timeline that I took for the actual science fair. And then for JIC, since JIC takes place in late October, I did definitely study a lot on my project over summer before going to JIC.
And then you kind of also talked, or at least touched on this a little bit, but when you talked about your mentors, but I wanted to kind of talk about the role that educators and teachers play in this. So what support from educators made the biggest difference in your journey?
I think for my science teacher and then science fair sponsor, even though they couldn't help me technically with all the simulations and tools, every day when I would come to school, they would check in. They're like, oh, hey, how's your science fair project going? Or when I go to the club, they would be like, hey, how's your project going along? If you need any help on the more bigger scale to look at things, or if you just need someone to look over your poster, someone to present to, I'll be there. And so my educators and my teachers definitely played a huge role. I remember presenting my science fair board to my teacher and science fair club sponsor before actually taking it to the fair and then actually taking it to JIC. So they were really helpful just to be there, even if they couldn't help on the project itself.
Thank you. I don't see any more questions coming in. Oh, we got one more, but I'm going to ask one more and then we'll go through this one before we wrap up if no more come in. So I know, so my question is, how can students get started if they don't have access to a lab or formal research program? And I know, like in your case, you're doing simulations. So I know that's one way to do it, but can you talk a little bit on that of, what that might look like for someone that, again, doesn't have access to a lab or a formal research program?
Yeah, without access to a lab or like a formal program, it's fine. There's definitely a lot of things to do online for simulations. The way I did simulations was I just found websites online, and what it might look like would be you would just try, and when you start to do your research plan and plan forward all the tools you're going to use, just search tools to do what you want to do. So I did tools to dock one protein to another, or tools to find the 3D structure of protein from an amino acid sequence, or like a website that can find the binding energy. And once I found a list of websites, I would go through and find articles on these websites and on these tools and see which ones were reliable and which ones weren't. And then I would write these tools down and kind of make my own research program in a way and figure out what I was going to be doing for the rest of the project.
Yeah, I think one thing that I'm hearing a lot, which, I want to emphasize, is that the internet is a very powerful tool. There's so many things out there. Google, search for what you're looking for, and I'm sure there's something that can at least get you started.
Yeah, definitely.
So we have a few more questions coming in, but we're going to go ahead and wrap up. If you don't mind, Pranshi, can we share your email or something like that for anyone with follow-up questions?
Of course. I'll just put it in the chat.
Perfect. And I will share it as well once I send out the recording and everything. So while we do that, I'm going to go ahead and wrap up. Before we conclude, I'd like to take a moment to thank our sponsors. Their generous support helps make these cost-free webinars and STEM opportunities possible for students and educators around the world. We are incredibly grateful for their commitment to expanding access to STEM education, research experiences, and innovation opportunities for the next generation of scientists and leaders. Last but not least, thank you all for joining us today and for being such an engaged audience. We truly appreciate your time and participation. A special thank you again to Pranshi for sharing her inspiring journey, research, and insights with us today. I'm going to go ahead and put a quick poll up for those that are here. It's just a quick survey on how you thought today's session was. So please take a second to fill out. It's be really quick, just 6 questions. And then for those that are watching asynchronously, the QR code is there. Please go ahead. And again, it only takes a few minutes to fill it out. Your feedback is very important to us and helps us improve future webinars and better support students and educators. So with that, again, thank you again to everyone, and we hope to see you at future events. Thank you. Yep, Pranshi, I'm going to leave this up for a minute just so the survey saves up, but you're welcome to log off whenever. Thank you again so much for being here, and I'll send you a follow-up email as well.
All right, yeah, thank you so much. Thank you to everyone for coming, and thank you so much for having me. I really enjoyed it.
Thank you.